Hey everyone, Dr. D here. In this video, we are going to be covering Chapter 14, Infection, Infectious Diseases for Microbiology 2420. So let's get started. So here we're going to be talking about how pathogens cause disease. And when pathogens are on your body, that is actually a type of symbiosis. And one common misconception about symbiosis in biology is that it's always a good thing, but that's not necessarily the case. Symbiosis simply means to live together. You could live together in harmony, or you can live together and cause one another harm, right? You can be toxic with one another. And that's exactly what happens with a pathogen. With a pathogen, the pathogen is usually a type of parasite. So take a look at this. Parasitism is a type of symbiosis. However, it's a type of symbiosis where one organism benefits. That would be the pathogen, because it's feeding off of you, right? And organism two is harmed. So you are harmed while the pathogen benefits. That is a type of symbiosis. Again, what most people think of symbiosis is when both organisms benefit. For example, organism 1 benefits, organism 2 benefits, they live together, they both benefit. This is actually a type of symbiosis called mutualistic symbiosis. Commensal is another type. This is when organism 1 benefits, organism 2 is neither harmed nor benefits. So for example, you know how a lot of organisms on your body are part of your normal flora? For example, you do have certain skin bacteria on your face, on your skin, on your hands, different types of staph epidermidis, etc. You have E. coli and bacillus, lactobacillus in your intestines, in your guts. Those bacteria don't necessarily harm you, but they're benefiting by being on your surfaces, right? So that a lot of these normal flora on your body, inside of your mouth, right? These are commensal. Okay, so just be aware that there are different types of symbiosis: mutualist, commensalist, parasites, okay? And all of them are types of symbiosis. Again, your normal microbiota on your body, this is your normal flora. These are usually types of commensalists. Okay, now these are organisms that colonize the body's surface without normally causing any disease or harm. A lot of times, though, these can be what are called opportunistic pathogens. So, for example, if I were to get a cut on my skin, those normally harmless bacteria, those normal flora on my skin, for example, Staph aureus, Staph epidermidis, these types of bacteria could get into that cut and now cause an infection. So that's what an opportunistic bacteria is. It's a bacteria that doesn't normally cause harm, but when presented the opportunity, such as a break in the skin, can then get in there and cause a disease, cause inflammation, cause problems, okay? So these are also termed normal flora or indigenous microbiota, etc. Now there's two types. There's the resident microbiota and the transient microbiota. What does that mean? Resident microbiota means these are the usual suspects. These are the bacteria that are 9 times out of 10 found on your body. They belong there. They outcompete other bacterium for that spot on your body. Transient microbiota may be something that you come into contact with, but it doesn't establish a foothold. It doesn't last very long. It usually passes through the body, passes by. Okay. So resident microbiota. These are part of your normal microbiota, your normal flora. For example, all the bacteria in your gut, all the bacteria on your skin. These are mostly commensal. Remember I said commensal, which is a type of symbiosis. And you can see some examples of resident microbiota. There are a huge number in the upper respiratory tract, a huge number in the digestive tract. These are just examples for you to look through and glean. These are not for you to memorize, obviously, but I just want to give you an idea of all the resident microbiota there are on your body. Don't forget, if we're not counting for red blood cells, there are 10 times more prokaryotic cells on your body, like bacteria cells on your body, and that includes inside of your guts, then you have your own cells. So your body is covered in microorganisms, okay? Your body has 10 times more bacteria on it, and that, of course, again, it includes your intestines, then you do have your own number of cells. So you're covered in these little guys. Okay, and some of them, like I said, are resident, and some of them are transient microbiota. Okay, and here are just some more examples in the urinary reproductive system, the male urinary reproductive system, the eyes, the skin. Everywhere you look that's on your body, you have microorganisms. So what do I mean by on your body? Do you guys remember how I said that humans are kind of like a donut, like an elongated donut? On your body means your skin, anywhere on your body, but on your body includes inside of your mouth, because that's still not inside of your tissues. Does that make sense? Like, so the inside of your mouth is still on your body. The inside of your esophagus or trachea is still on your body. The inside of your lungs is still on your body. Your stomach, the inside of your stomach is still considered on your body, and your gut tube, your upper and lower digestive tract. All of that is technically not in your body, it's on your body. It's not inside of your tissues, right? And remember what I said in class, you are full of bacteria on your body, but not in your body. There should not be any bacteria or viruses in your body. What do I mean by in your body? I mean inside of your tissues, inside of the actual bloodstream, inside of the actual muscles, inside of the actual tissues. Does that make sense? So you shouldn't have the presence of microorganisms in your bloodstream. You shouldn't have them in your tissues. That is indicative of an infection, okay? But your body is covered in microorganisms outside of the tissue. So what do I mean by that? That includes this on top of your skin, inside of your mouth, inside of your stomach, inside of your intestines. That's all. And you're inside of your colon, right? All of that is still not technically inside of your tissues. Remember, because we are a giant donut, right? The inside of the donut, the whole of the donut still outside of the donut, if that makes any sense. So anyway, let's keep going. So remember, you also have transient microbiota. Remember what I said? These are bacteria that remain on the body for a short period of time. Why don't they get a foothold? Well, maybe they don't attach to your cells very well. Maybe they don't attach very well. Maybe they can't grow on the salty skin so well. Maybe they can't grow in the low oxygen conditions of the intestines very well, right? So they don't persist in the body for long. And sometimes they're even eliminated by the body's defense system, okay? And like I said, salt or peristalsis of the esophagus, things like this, or the intestines, all this stuff can cause shedding of these organisms and your body to rid these organisms after a short period of time. So where did you get your microbiota? Well, when you are developing as a fetus in the womb, the embryo grows inside of the womb of the female. These fetuses are free of microorganisms. So if you're pregnant and there's a baby inside of your womb, right? That fetus is free of microorganisms. It's not until the birthing process, when you're giving birth and you're exiting the birth canal, right? That's when the baby comes into contact with the mother's skin, right? With the mother's other tissues, And that's when they pick up their first normal microbiota. Does that make sense? So during the birthing process, that's when the otherwise sterile, you can consider it sterile, fetus picks up its normal microbiota, and a lot of those become its resident microbiota that stick with the child for the duration of its life, along with other bacteria it picks up, shortly after birth as well. But much of one's resident microbiota are established during the first months of life. So during your first few months of being, born, that's when you pick up your resident microbiota. Your body gets covered in all these bacteria that are going to be kind of with you for the rest of your life. You'll never be a microorganism free again, right? Until after death. But how do these normal microbiota become opportunistic pathogens? So, what does opportunistic pathogen mean? Normal microbiota, like the bacteria that exist on your skin, that can cause disease under certain circumstances. Well, what if you become immune suppressed? What if you introduce those bacteria where they're not normally found? Like in the urinary tract, for example. If bacteria go into the urinary tract, you could get a urinary tract infection. If you become immune suppressed, you could get an infection. If let's say there's a cut in your skin, that could cause an infection. Okay, that's another example of unusual site in the body. Changes in the normal microbiota. So if you somehow were to destroy the normal microbiota and then change the ratio of the microorganisms on that tissue, that might cause an infection as well. All right, so how does the infection work? Well, there's a host. If we're talking about human diseases, then the person is the host. The host means the larger organism that supports the survival and growth of the smaller organism. And then the parasite is the organism that grows on the host. It has to do what? The parasite must live on or within the host organism and become metabolically dependent. That means feed off of the organism on the host. And this causes disease, right? Any organism that causes disease is a parasite. And why do they cause disease? Because of feeding off of the organism, causing damage to the tissues and cells of that organism. This is what we're talking about when we're talking about host-parasite relationships. So what is an infection? Here's just some terminology I'm going to rattle off, but it's important to understand this terminology. Infection. What does infection mean? A parasite grows, becomes, metabolically dependent on the host. Parasite grows, multiplies within or on the host, and then this can cause disease. It may not be an overt disease, but let's talk about different types of disease types. And we'll talk about that in a minute. So the pathogen, any parasitic organism that causes infectious disease. It could be a frank pathogen or primary pathogen, which causes disease by direct interaction with the healthy host, or an opportunistic pathogen. Remember, this is part of your normal flora that can cause disease when it gains access to somewhere where it shouldn't be or the host is immunocompromised. So frank pathogens, for example, Bacillus anthracis, anthrax, right? the bacteria that causes anthrax, that's a frank pathogen. You come into contact with that bacteria, you're going to get the disease. Whereas, Staphylococcus epidermidis, that's an opportunist. That's something that lives on your skin. And unless you give it reason to, it's not going to cause any problems. You see what I'm saying? The big difference there. What's pathogenicity? Pathogenicity is the ability of a parasite to cause disease. So, for example, again, Bacillus anthracis, the bacteria that causes anthrax, has a high pathogenicity, whereas Staphylococcus epidermidis would have a lower pathogenicity. You would have to introduce it to a site where it otherwise shouldn't ever be for it to actually cause a disease. Does that make any sense? Now, once the the parasite is on the host, what can happen? Well, there's a latent state. This is the state in which there's no transmission of the organism. There's no transmission of the organism. No symptoms are present. So this is kind of like a hibernation or dormant phase of this organism. Examples are when you have herpes infections, when it goes dormant. There's no transmission. You can't pass it on to anyone else, but it is still present. Tuberculosis can go dormant. Leprosy, you can't pass it on, and there's no symptoms yet. All right, there's no symptoms going on. That's called the latent stage. All right, so before I get into the infectious disease process, which is really interesting, actually, it's very important to understand these terms as a medical professional. Let's take a quick break time with Tig, and we'll be right back. Hey everyone, welcome back. Let's get started. So, during the infectious disease process, what you need to understand is that there's deterioration of the state of health due to the pathogen, the virus, the bacteria, the fungi, protozoa, helminth. And what you really need to understand when you become a healthcare professional is that there's a big difference between the term signs and symptoms of the disease syndrome. Disease syndrome means the set of characteristic signs and symptoms of the disease. So what is a sign? A sign is an objective change in the body that can be directly observed. So for example, if I have the flu, the signs would be the fever, the cough, all of the things that the doctor could measure. Does that make sense? All of the things the doctor could objectively see, measure, determine. Let's say if I couldn't talk, right? I can't talk. What can be determined by the doctor? Let's say if you have a pet, like your cat or dog. If you take your cat to the vet, the signs would be whatever the vet could determine about what's wrong with your cat. Does that make sense? Because your cat can't talk. Your cat can't say, oh, I'm not feeling very well, right? Your cat might behave in a strange way, but it can't tell you anything about what's going on inside. Signs are things that are measurable. Does that make sense? Symptoms, on the other hand, are subjective changes experienced by the patient. So let's say if I have the flu, the symptoms would be fever. Of course not fever. The symptoms would be fatigue, right? Does that make sense? The symptoms would be fatigue. You know what fatigue means? Feeling of tiredness, right? Is there a way for the doctor to measure tiredness or determine if I'm tired? No. If I'm feeling fatigue, or let's say I'm feeling nauseous, I feel like I might throw up, there's no way for the doctor to know that unless I tell the doctor that I'm feeling nauseous, right? Does that make sense? So there's a huge difference between signs and symptoms. Even pain is a symptom, right? If I'm feeling pain in my shoulder, doctor had no way of knowing that, right? I had to mention, you know what? There's pain in my shoulder. So pain is a symptom as well. Does that make sense? So please be careful when you become a healthcare professional later on down the road, if that's your goal, that you're being accurate when you describe signs and symptoms, right? Signs can be measured, symptoms cannot. They have to be expressed, right? Very good. So now let's say there's an infection, right? What happens during the course of the infectious disease? There are different periods or stages, right? There's the incubation period, prodromal stage, period of illness, and then convalescence. This is what happens with diseases. The first part is called incubation period. This is when you've come down with a pathogen. The pathogen has entered your body, but before signs or symptoms appear, right? Let's say I pick up the flu. The flu doesn't make me sick immediately, does it? No, it takes a few days, right? So that period between when I picked up the flu and before I start feeling weird, that's called the incubation period. The prodromal stage is when I have onset of signs and symptoms, but it is not clear enough for diagnosis. So what does that mean? Let's say, you ever caught a cold, right? And then you kind of wake up one day and you're like, "Ooh, it hurts to swallow, like my throat is all hurt and itchy and weird. It hurts when I swallow. Oh gosh, something scratchy back there. Man, I don't feel good. I think I'm coming down with something." That's the prodromal stage. But if you went to the doctor right then and there, the doctor may or may not know what's wrong. The doctor might say, "Well, sore throat, you could have a cold coming on, or maybe it's some kind of strep throat, or maybe it could be this. There's not, it's, because you don't have the full-fledged illness, the doctor cannot, diagnose. The doctor could guess. The doctor could say, well, it could be this or this, but it's not until the period of illness. This is when the disease is the most severe, right? And has its characteristic signs and symptoms. That's when the doctor can truly diagnose you, right? So let's say it's not just itchy throat. You have the full-blown cold, which means you're coughing all over the place. You're blowing your nose is runny. Everything's like you're feeling tired. You have all the signs and the symptoms of the cold, right? And then at some point, hopefully, you fight off the cold, and then you enter convalescence. This is also called senescence phase. This is when the signs and the symptoms begin to disappear. They go away because you're recovering, right? So here it is plotted. These is those parts of the infectious disease all plotted out. You can see here on the y-axis, they've plotted intensity of the symptoms, so how bad you feel, right? And on the x-axis, they've plot time, right? So here at the beginning of time, that's when you picked up the virus, you picked up the bacteria, you picked up the pathogen. Remember, for a while, do you feel bad? Do you feel symptoms? No, you don't feel anything at all for a little while. That's called incubation period. From the time when you picked up the flu until the time when you start feeling any kind of symptoms at all, or showing any kind of signs. And then at some point you wake up in the morning and you're like, "Oh, my throat's itchy." Remember, that's the prodromal stage. Prodromal stage, you start feeling bad, but it's not clear enough because the signs and symptoms have not presented themselves enough to be accurately diagnosed. That's called the prodromal stage. And then you start really feeling bad, right? You start really feeling bad. This is the illness phase. This is when you show all of the telltale signs and symptoms of the disease. And then at some point you max out your suffering, right? That's like the worst it's going to get. This is when you're in the throes of it. And then at some point, your body takes charge, your immune system fights off the infection, and you start feeling better, right? The signs and symptoms start to abate, and you recover, hopefully. This is called the convalescence period, okay? So this is how it works. This is the course of an infectious disease. Now, where did you pick up the infection? Where did you pick up the pathogen? Well, that's the source. The source is the location from which the pathogen is transmitted to the host. Where did you pick up that pathogen? The period of infectivity is the time during which the source is infectious or is disseminated the organism. So, if a person is infective, if the person is infectious, if the person can cause dissemination, that means release of the virus, release of the bacteria, that's the time of the infectivity. So people ask, like, am I infectious when I am feeling better from the flu? Can I still make others sick? That would mean it's still in the period of infectivity. Reservoir. What does the reservoir mean? It's the site or natural environmental location in which the pathogen is normally found. It's sometimes the source of the pathogen. So the reservoir means, where do you go to find the pathogen naturally? Where's the natural habitat of the pathogen, right? The source may or may not be the reservoir. Does that make sense? I want to quickly talk about the difference between source and reservoir. Source is where you picked up the pathogen, but it is not necessarily the reservoir. The reservoir is where it's normally found. So let me give you an example. Okay, let's give you a quick example. All right. Let's say you're eating at your favorite sushi place. Okay, you're eating at your favorite salad or sushi place, and then you become really sick with, like, oh, my intestines hurt, my stomach hurts, I have food poisoning. Oh gosh, I got food poisoning. Now, what was the source of that food poisoning, do you think? Where did you pick up that pathogen, do you think? Yeah, probably from the salad or the sushi that you ate, right? That's the source. But does that mean that salad is where that bacteria, let's say it was some bad strain of E. coli, does that mean that salad is just a normal place to find E. coli? Or sushi is a normal place to find E. coli? No, that's where you picked up the E. coli. Does that make sense? The E. coli probably came from one of the people who handled the food, maybe the chef or someone who prepared the food, didn't wash their hands, because guess what? E. coli is a gut resident, right? E. coli lives in the intestines, right? So if the chef went to the bathroom and didn't wash their hands, well, that's called fecal contamination. And then the reservoir would have been the person's intestinal system, right? The person's digestive system. The person touched the food and made the food contaminated, and now the food is the source, but the food is not normally the reservoir. Does that make sense? So again, if you pick up a bacteria from a doorknob, that's a source. That's not a reservoir. The source is simply where a person picks up the pathogen, but the reservoir is where it normally resides. Okay, so just be aware of that. And what is a carrier? A carrier is an infected host who is a potential source of the infection to others. So I might have the flu, and I might seem fine, and I might be able to still spread the virus to others, right? That's called a healthy carrier. Healthy carrier harbors the pathogen but is not ill. Incubatory carrier is someone very similar to healthy carrier, but is someone who's incubating the pathogen in large numbers but is not yet ill. So this person's going to become ill, but they're not yet ill. They're spreading the disease. Healthy carriers are someone who may or may not even get ill, but spread the disease. They also call these healthy carriers, they call them, you guys remember, asymptomatic carriers, right? Now with COVID, you've heard of these asymptomatic carriers. These are otherwise healthy people who are roaming around, but spreading that SARS-CoV-2, that COVID virus. They're spreading it to other people, but they seem healthy. Those are called asymptomatic carriers. Convalescent carrier could be someone who has recovered but continues to spread the pathogen, or an active carrier. This is someone who's constantly coughing and is definitely has the overt clinical case of the disease and is definitely spreading it to others. And again, we need to understand carriers. If you're in the healthcare profession, you need to understand something, and that is that just because you look healthy does not mean you're not a source of pathogen to others. It does not mean you cannot make others sick. And this is something that the general public is starting to become aware of with this COVID-19 that's going around, right? I might seem perfectly healthy, but I might be just disseminating SARS-CoV-2, the virus causes COVID-19. I might be spreading that all over the place like gangbusters, and I might seem just fine, right? Again, that's because I'm a healthy carrier, for instance. Someone could have just gotten over the flu, right? And still be spreading the virus. That's a convalescent carrier. Someone could have picked up the flu or picked up COVID, SARS-CoV-2, and they might be spreading it. So what you need to understand is just because someone's not coughing their lungs out, right? Just because someone's not sneezing all over the place, doesn't mean they can't spread the disease to other people, because you could be a convalescent, healthy, or incubatory carrier. Does that make sense? And that is why they recommend wearing masks nowadays with this quarantine, with the opening up the opening up all the stores again. You got to wear a mask even if you're feeling well, not necessarily to protect you, but to protect others, because you might be infecting others and not even know it. Or you might become infected by someone who seems just fine, right? This is what people need to understand, right? You're not safe just because you're hanging out with people that seem fine, right? Why? Because there are different types of carriers, and definitely not all carriers look sick. Okay, very important thing to understand, and the general public is starting to understand this. Now, what about pathogens that you pick up from animals? These are called zoonoses. Okay, so when you see this term zoonoses, you should know that it is a pathogen that transmitted from an animal to a human. And a lot of times it's not a direct transmission. It's not like you encountered the chipmunk and the chipmunk gave you the disease. There could be these intermediaries, these arthropod vectors. Do you know what an arthropod is? It's an insect or a arachnid or some kind of biting organism, something like a mosquito, a tick, a mite, a bed bug, something that, let's say the tick bites a deer, right? The tick bites a deer, and then the tick bites the human. These are called vectors. Let me tell you that term real quick. Vector means a living transmitter of disease. So a mosquito is a great vector. It's a living organism, and it transmits all kinds of viruses, all kinds of bacteria, all kinds of protozoa, like protists. Arthropods are these biting insects. Vectors are living transmitters of disease. So a lot of times when you catch a virus, it might be from a mosquito who bit a bird, for instance. Does that make sense? Now, let me show you an example of a zoonoses. Zoonoses means a pathogen that's spread by animal to human. Let's talk about the avian flu, bird flu. This is a flu virus that normally infects birds, such as ducks. That duck would be the source of the pathogen. It could be a healthy host, a healthy source. A person could handle the source. A person could handle the duck. And guess what? The duck spreads the virus, spreads the flu to the person. This person may or may not become sick. In this case, it looks like the person is a healthy carrier. And that healthy carrier might be able to spread the disease but not become sick. But what happens when the healthy carrier handles a toddler, someone with a more susceptible state, someone with a more susceptible immune system, less robust immune system? Well, then the child becomes sick. So now the child caught the flu, right? So you can see how this chain of events works, this chain of events. These are zoonoses, transmissible from humans to animals. This is why we should limit our exposure, close exposure to living animals. How else can we transmit the infectious disease from, one source to another? Well, there are direct examples of transmission and indirect examples of transmission. What are direct? Direct include things like horizontal contact, kissing, sex, for example, airborne droplets, people who spray it instead of say it. They're just splattering, they're talking with such emphasis that they're just spraying their little droplets everywhere. Vertical contact, this means through the birth process, from mother to daughter, mother to son, vertical contact. Or vector. So for instance, look, these flies may have landed on dung and then come and landed on the person, or these flies, some other way of transmitting directly to the person, right? Now, indirect examples. Indirect examples can include the contact surfaces such as fomites. Do you know what fomite means? It means a surface, like a doorknob, right? A doorknob, a bed, a couch, anything that you can touch, right? Anything that you can touch. A lot of times fomites are also called vehicles, inanimate objects that you can touch and pick up microorganisms from that, pick up the pathogen from that surface. So why is this an indirect example? Well, someone else touched the doorknob, then you touched the doorknob. Does that make sense? That's indirect. That's not like kissing someone directly, right? Or spitting on someone directly. Does that make sense? So that's why it's indirect. How about preparing food? This chef didn't wash her hands, right? This chef did not wash her hands. She touches it looks like sushi. She touches the sushi. She didn't wash her hands, so maybe she had some E. coli on her hands from using the bathroom, right? The normal source, I should say the normal reservoir for E. coli was her intestines. And then she didn't wash her hands, so the E. coli is now transmitted to the food. The food becomes the source, right? And then someone else has to eat the food. So you see that was indirect right there. And then airborne. So do you remember that when you speak or talk, I've said before that you release a mist, a very fine mist, and these are called droplet nuclei. And this mist can linger in the air for hours, sometimes days, and that's an example of indirect. Someone could be talking in the room and leave the room. You enter the room hours later, you could become sick from the droplet nuclei. Same thing with the dust. Dust could harbor pathogens as well, and it could linger in the air, and then you can come in contact with that dust and become sick. So, again, pathogen transmission could be when the pathogen goes to host. That's what that means, pathogen transmission. So initially, the pathogen goes to the host becomes sick, gets the disease, and then the transmission can go from that host to another host. So this is disease spread. The pathogen goes to a host, that host becomes sick, that host passes it to another host. But what you need to understand is transmission by itself does not make for the infection, right? The pathogen must make contact with the appropriate host in the tissue. This is tropism. Do you guys remember that viruses, when we talked about viruses, this concept of tropism came up? It's very important for you to understand that not all pathogens can infect you. Not all pathogens can infect all of your tissues. For example, we're talking a lot about SARS-CoV-2 nowadays, right? Again, the virus that causes COVID-19. What is its tropism? What is its tropism, you guys? Do you remember? Can it infect your skin? Can SARS-CoV-2 infect your skin? Can it infect your reproductive organs, right? Genitalia? No. It can infect what? It can infect the upper respiratory tract, right? The inside of the soft palate of your throat. It can infect your lungs, your upper and lower respiratory tract, right? Yeah, it can, right? And I've heard it can infect the eyes as well. Okay, so that's the tropism of SARS-CoV-2. This is why you wear masks, right? Because you don't want to get the virus into your lungs. If the virus lands on my skin, big deal. I just washed my skin. I'm not going to get infected. I could touch the virus and then wash my hands with soap and water for 20 seconds and it's gone. It didn't infect my skin, did it? But what you need to understand is that's because of the tropism of that virus. That's the particular tissue that gets infected by the virus. And usually it's because that cell, so let's say my lung cells have particular receptors. These are proteins usually on the surface of the cell. And these receptors are required for the virus or the bacteria or whatever pathogen to attach to the cell. Does that make sense? So in the case of COVID-19, for instance, your lung cells have a surface receptor called the ACE2 protein, right? ACE2. And without that surface receptor, without that surface protein ACE2, the virus cannot attach to your cells. And it's because your lung cells have this ACE2 protein. That's why the virus infects that cell. Now, before we get to other pathogen transmission mechanisms, let's take another quick break with Tig. I'll refresh my water bottle. All right, guys, welcome back. Let's go ahead and start with pathogen transmission, different modes of transmission. Let's talk about airborne transmission first. Remember, you can pick up a pathogen from the air. For example, if somebody is coughing or sneezing, look at this. There are bigger droplets, and then there are fine mist-like droplets. The bigger droplets are just simply called droplets, but the tiny particles, these tiny droplets are called droplet nuclei, and these are very small particles. We're talking 1 to 4 micrometers in diameter. They're so small that they can kind of drift around. They can remain airborne for a long time, hours to days. They can travel long distances. They usually are propelled from the respiratory tract through sneezing, coughing, even vocalization, even talking. So this is why respiratory diseases spread so easily. It's because when someone coughs or sneezes, they can form these droplet nuclei. These droplet nuclei are quite small and they could harbor the pathogen. The pathogen can then remain airborne for a long time and travel a long distance. Also, dust, right? Dust particles are an important route of airborne transmission too. This is why we have to incubate our plates in the lab upside down. This is why, because dust particles can travel a long distance. Okay, and when dust settles on a plate, when dust gets inhaled by your lungs, for instance, then you could get an infection. You can become infected by the pathogen. Now let's talk about contact transmission. Remember, there's direct contact, person to person, as we talked about before with kissing, touching, sexual contact, and indirect contact, which involves an intermediate, sometimes an inanimate object. Remember, a fomite or sometimes called a vehicle, eating utensils, beddings, doorknobs, etc. What's a vehicle? A vehicle, like I said, is an inanimate object or material. You could sometimes call these fomites. These are vehicles. So, for example, water could make you sick, but not because water is by itself dangerous, but because it could be contaminated, right? Same thing with food. Food could make you sick because it could be contaminated. Surgical instruments, bedding, eating utensils, doorknobs, these are all vehicles, inanimate objects that could harbor the pathogen and transmit the pathogen to the human. And what's the difference between a vehicle and a vector? Remember, vectors are living transmitters of disease, such as arthropods. Arthropods include mosquitoes. Mosquitoes are a huge bane for humans, right? They harbor so many different diseases, and they transmit those diseases to people when they bite or when they defecate near the bite site, or, a number of ways that they can transmit that disease. Now, a lot of times you don't become sick simply by encountering one virion or one bacteria, one pathogen. You need what's known as the infectious dose. What's the infectious dose? The infectious dose means the amount of viruses that will usually make you sick. The infectious dose 50, you see this term ID 50? That means infectious dose 50. That means the number of pathogens it will take to infect 50% of an experimental group. It varies with the pathogen. So if I encounter one E. coli, bad strain of E. coli, I probably won't get sick. But if I encounter 50 million, then that might be right where I start becoming sick. That might be the infectious dose 50. So how many E. coli does it take for me to ingest? And this would be bad strains of E. coli. How many bad strains of E. coli would I have to ingest? 1 E. coli, 50 E. coli, 50 million, 50 billion E. coli, 50 trillion E. coli? How many do I have to ingest for half of the number of people to become sick, right? That's the infectious dose 50. For example, the infectious dose really is important because it could be the difference between becoming very sick and not very sick, or not sick at all. So one of the big concerns about, for example, healthcare workers during this COVID-19 outbreak, one of the big concerns is infectious dose. Let's say you're a nurse and you're working in close proximity with COVID-19 patients. Well, what if someone coughs really, really bad and you're right next to them and you breathe in millions and millions of virus, right? You just inhaled a big infectious dose. Whereas if I go to the grocery store and someone kind of coughs off in the corner of the grocery store, I walk by a little bit later and I breathe in something, well, I picked up maybe a few dozen of the virus, So my infectious dose being at the grocery store was very little compared to the infectious dose received by the unfortunate nurse who was working with the COVID-19 patient right in front and inhaled, a cough, right? That could cause worse outcomes for the nurse, right? Because the nurse was given a bigger infectious dose. That could cause a bigger insult, a bigger disease, right? A faster ramp up to the disease. And that could cause the nurse to become much more sick than if she had, picked up the virus from a grocery store in a lot less concentration, or a lot less number of virus, right? So it's very important to understand the infectious dose, right? How many pathogens did you encounter when you got sick? Did you encounter just a few dozen? Well, then you probably either won't get sick or you're not going to get very sick at all. Did you encounter a few million, right? This all makes sense. Growth rate. Now, the pathogen must be able to find a favorable condition to grow in the host, right? Extracellular pathogens simply means that the pathogen can grow outside of the blood, outside of the cells, I should say, outside of your cells. So in the fluids of the body. Extracellular just means the pathogen does not need to invade your cells to grow. Intracellular pathogen means that the bacteria or the virus or what have you must infect your cells in order to multiply. Now, let's talk about host susceptibility. How susceptible are you to a pathogen? Well, it depends on two main factors. The defense mechanisms of the host. Is your immune system robust? Is your skin intact? Are your mucous membranes working properly? Right? How healthy are you? All of that factors in. And the pathogenicity of the pathogen. How dangerous is the pathogen? If the pathogen has a high degree of pathogenicity, well, then it's going to make you sick regardless of your immune system. Does that make sense? Also, what factors into your defense mechanisms of the host? What factors in? Well, nutrition. Are you malnourished? Do you have issues with your nutrition? Genetic predisposition. Some people are just more genetically predisposed to particular pathogens. Stress. Are you really stressed out? I could add sleep on here. Are you not getting enough sleep? All of these things can play a role in the host susceptibility to infection. So, for example, if you have poor nutrition, you have a predisposition to coming down with a particular type of virus, and you're stressed out and not sleeping very well, well, that's just, the pathogens are going to have a field day, right? They're going to have an easy time causing an infection. Now, once the host has been infected, the pathogen must exit the host and find new hosts, right? So it has to spread to other hosts. Now, this can happen in active pathway, active escape, or it could be a passive mechanism, passive escape. Active means that the pathogen actually moves to a portal of exit, whereas passive means it piggybacks on something else, such as feces, urine, droplets, blood, saliva, desquaminated cells, shed cells, etc. So I'm going to give you an example of these real quick. So, for example, if I go to the bathroom and don't wash my hands, right, and I touch food, and that food now grows bacteria and gives someone food poisoning, which one was that? That was an example of passive escape, right? Active escape would be the pathogen itself makes its way to a portal of entry. For example, have you guys heard of the guinea worm? This is a worm that infects your body and then bores its way to your skin, usually in the lower extremities, the foot, the shins, right, the calves, and then it pokes its head out and it actually exits it, and it disseminates its eggs, right? So if the pathogen moves itself to a portal of exit, that's active escape. If it piggybacks on blood or urine, feces, etc., then it is passive escape. Virulence. Let's talk about virulence. What does virulence mean? Virulence means the degree or intensity of the pathogenicity. And what does that depend on? Remember, pathogenicity means the ability of a pathogen to cause a disease, right? But what is the degree or intensity of that pathogenicity? That's virulence. And virulence depends on certain virulence factors. Virulence factors are things that determine the degree to which the pathogen causes damage, is invasive or infective. It could be a number of things. There are a number of things that can contribute to virulence, right? And virulence factors include a number of different things. I'm going to give you some examples. So, virulence factors could include things that give the ability to adhere, right? If do you guys remember the structures like slime layers or fimbriae? These things allow for adherence, right? So fimbriae could be virulence factor. Slime layer could be virulence factor. Why? Because those structures enhance adherence of the pathogen, the bacteria, to the host. Colonization. So the ability, anything that gives you the ability to colonize, to grow on the host, that's a virulence factor. Anything that allows you to invade the host, that's a virulence factor. You see, so all of these things are virulence factors. Now, the first step of an infectious disease is to enter and attach to the host. If you can't enter the host, if you can't attach to the host, that's the first step. You're not going to cause disease in the host. So what are the portals of entry? The skin, respiratory tract, gastrointestinal. There's a number of different ways. This can be due to a vector. A vector could cause it. Sexual contact could cause it. Blood transfusions, organ transplants, all of these can cause entry. Now, adherence. Remember, this is when the bacteria or virus, whatever, this is when the pathogen attaches to you, attaches to your cells, your tissues. This is mediated by special molecules called adhesins. Adhesins are structures that allow the organism to attach to your cells. Structures like pili, fimbriae, adhesion molecules, slime layers, things like that. All of these are virulence factors. All of these are virulence factors. Why? Because without the fimbriae, would the bacteria attach to you? No. Would the bacteria cause an infection? No. So fimbriae themselves are virulence factors because they contribute to the virulence of the pathogen. Does that make sense? And then what's the next step? After adherence, you have to colonize. You have to establish a site. You have to establish a place where you could grow. If you can't grow on the host, you're not going to cause a disease. Does that make sense? Now, once you've done all that, you want to invade. You're gonna invade the organism. So infectivity means the ability of an organism to establish discrete focal point of infection. Once you form that focal point, you need to be invasive. You need to spread to other tissues, adjacent tissues. You could, penetrate the mucous membranes or the epithelium. That means the cells, the tissue cells, right? This could be active or passive. Again, let's talk about active penetration. Active penetration. This could be through lytic substances. That means substances that break your tissues, break apart your cells, break apart your tissues, allow the organism to squeeze between the cells of the tissues, etc. This could be passive. This could be a skin lesion, for instance. Let's say I have a cut. I get a cut on my skin. That's going to allow the bacteria to passively infect me. The bacteria didn't have to break through my cells by releasing enzymes or something like that. The bacteria was able to simply go past my skin because I had a skin lesion. Insect bite. This one is definitely on the test, and this one is definitely one that students get confused. Insect bites are an example of passive penetration of the pathogen. Why? Because did the pathogen itself penetrate your skin, or did the insect penetrate your skin? The insect penetrated your skin, and then the bacteria entered the skin to your tissues. So the fact that the insect bit you does not make it an active invasion. It does not make it an active penetration. It's still passive because the pathogen itself didn't have to do anything to pass your skin barrier. The insect did, if that makes any sense. Wounds. Okay, if I have a bad burn, for instance, that could allow for passive invasion. Now, once in the circulatory system, remember, should viruses be in the circulatory system? No, but once they do, this is bad. This means that you have a bad infection, right? And so if there's bacteria in your blood, which there should never be unless you have an infection, this is called bacteremia. If you have bacteria or other pathogens or toxins in the blood, this is called sepsis or sepsemia. If you have a virus in your blood, that's viremia. So depending on what's going on in your system, there should not be any pathogens like bacteria, viruses, pathogens, protozoa, the worms. There shouldn't be any of this stuff. Fungi. There shouldn't be any of that stuff in your blood. But if there is, that's an example of sepsis. Sepsis would be any of these players in the blood. Or you could be more specific and say there's bacteria in the blood, viruses in the blood, worms in the blood, etc. Now, how do you resist host defenses? Let's say, so the bacteria now gets into the host. Well, guess what? The host has a immune system, doesn't it? And the immune system can destroy invaders, can't it? Well, hopefully that's the thing. Some bacteria can avoid the immune system, right? Some bacteria, some viruses can destroy the immune system itself. Like look at HIV. HIV infects helper T cells. HIV can destroy those helper T cells and cause major damage to your immune system, the very system that's set out to rid your body of the virus, right? So there are many ways that bacteria or viruses or other pathogens can evade your immune system. Okay. So one such example, lengthened O side chains. They could eject themselves from hosts to find more hosts. They could build decoy proteins so that they keep the immune system preoccupied. They could grow in biofilms. Do you remember what a biofilm is? It's like a community of different bacterium growing inside of a substance, a gooey, proteinaceous, sugary substance, right? And remember, bacteria that are inside of biofilms, it's hard to treat them with antibiotics. White blood cells can't penetrate the biofilm, etc. Now, the last concept of the chapter is toxins. Let's take another quick break time with Tig, and we'll get right on that. Let's talk about these toxins, which is another type of virulence factor. All right, guys, we'd say we finish off this chapter. So we were about to talk about toxins. These are toxins that bacteria produce, okay? And toxins are another form of virulence factor. There are two forms of toxins you need to know about, exotoxins and endotoxins. So let's get started. Before we get too deep into what exo and endotoxins are, let's just talk about the terminology involved with toxins. A toxin is a substance that damages a host. And it's usually what we're talking about is bacteria that produce these substances that damage the host. Toxogenicity is the ability to produce toxins. So a bacteria that has toxogenicity is a bacteria that is able to produce a toxin. Intoxication is a disease that results from the entry of that toxin into the host, and toxemia would be the condition of having toxins in the blood. Anything emia means in the blood. Now, what are exotoxins? Exotoxins are soluble, heat labile proteins. Labile means can be broken down. So these exotoxins can be broken down by heat. Why? Because they are proteins. And do you guys remember from Biology 1406 that proteins are these structures that have to fold correctly into what is known as a tertiary structure? And that tertiary structure is held together by relatively weak side chain interactions. So if I were to heat up a protein or change its pH, do you remember that would cause a protein to denature? That would cause a protein to unfold. So by heating up these proteins, like boiling them, for instance, well, then they're going to unfold and lose their toxogenicity. Okay. Now, usually these are released. So these are proteins that bacteria release as they grow. Most of the exotoxin producers are gram-negative bacteria, but not all. Okay, and they float around, right? These proteins float from one site of infection to other tissues. They can move around in the blood or other fluids of the body. So where do they come from? Well, they're synthesized by bacteria that have toxin genes. Remember, where do proteins come from? Proteins come from genes, a toxin gene. So a bacteria, for example, the bacteria that causes dysentery, Shigella, Shigella would have a gene called the Shiga toxin gene. And that gene codes for the Shiga toxin. The Shiga toxin is an exotoxin that causes dysentery. You see what I'm saying? So genes code for proteins. Proteins have functions, and the function of these genes, these toxin genes, is to form a toxin protein. So these genes can be found in the genome, or on plasmids, or on prophage. There are different examples of these gene locations. They are highly immunogenic. They are very lethal. What does immunogenic mean? Immunogenic means they can trigger the immune system. They can be recognized by the immune system. So what you need to know about exotoxins, remember, they are heat labile. They are proteins. They are coded for by genes, okay? They are coded for by genes. And what else do you need to know? They are immunogenic. What does that mean? Easily recognized by the immune system. They trigger the immune system to take action. So if these exotoxins are in your body, your immune system responds. Does that make sense? And what does your immune system do? Your immune system will try to neutralize those toxins by producing antibodies. Your immune system will make antibodies against that toxin. Okay. In fact, some vaccines, just a fun fact for you, some vaccines are actually inactivated forms of the toxin. These are called toxoids. So for example, the tetanus toxoid is an inactivated, chemically inactivated form of the tetanus exotoxin. When they inject this toxoid into your body as a vaccine, your immune system will make the antibodies and remember that protein in the future. So if you get a real Clostridium tetani infection, and that Clostridium tetani bacteria forms tetanus toxoid inside of your body will immediately neutralize it with the memory, which we're going to talk about in chapter 16. We're going to talk all about the adaptive or adaptive or specific immune system, the type of immune system that remembers these insults, remembers these types of infections, and forms your immune system, your white blood cell immune system. We're going to talk a lot about that stuff. All right, so there are different types of toxins. Obviously, some of the toxins target nerve tissue, like Botox from the botulinum bacterium. Clostridium botulinum forms a toxin called Botox, or botulinum toxin, targeting nerve cells. This is the same substance people inject into their face to reduce wrinkles and whatnot. This is a type of neurotoxin because it's a toxin that affects the neurons. There are enterotoxins. These are toxins that target the digestive tract, such as cholera toxin. And there are cytotoxins. These are toxins that generally affect tissues. They can affect all tissues, like diphtheria toxin. So, moving on from exotoxins to endotoxins, what is an endotoxin? Well, an endotoxin is very interesting because it is actually not produced as a protein. It is not a protein at all. In fact, it's a component of the gram-negative cell. You know how gram-negative cells have an outer membrane and an inner membrane, the plasma membrane? Well, Remember, the outer membrane of a gram-negative cell is made out of a structure called a lipopolysaccharide. Do you guys remember this? We talked about this in chapter 3, I believe. Lipopolysaccharide, LPS. And do you remember that part of the LPS is called the lipid portion of LPS, lipid A? So again, what is lipid A? Lipid A is a lipid. It's the lipid portion of LPS, lipopolysaccharide. And what is LPS, lipopolysaccharide? It is a major component of the outer membrane of gram-negative cells. And when this lipid A sheds from the cell membrane into the body, it's very, very toxic. It's very, very toxic. Now, the problem is, do you guys realize that lipids are relatively heat stable, not labile, right? So these endotoxins are stable. By the way, the only endotoxin is lipid A. There is no other endotoxins. When someone says endotoxin, they are talking about lipid A. Lipid A is heat stable. It's very toxic, and it's weakly. Here's the big problem with it. It is very weakly immunogenic. Remember what immunogenic means? It means triggers the immune system to take action. Well, because it's a lipid, it is not very well recognized by the immune system, so the immune system doesn't mount a very strong defense. So it is weakly immunogenic. So your immune system is not going to form antibodies very well at all. Okay, and it can get into if it gets into your system, it can cause sepsis, it can cause fever, weakness, diarrhea, inflammation, a host of other problems. It's very toxic, basically, to your system. It can even cause death in high enough concentrations. And by the way, when you have all this toxin in your body, this can lead to septic shock. This is basically when your blood cells are not transporting enough oxygen to your body. You've got too much toxin inside of your body, and you die, right? So sepsis. Just making sure I didn't. By the way, do you remember we talked about the hypothalamus before? We talked about how presence of toxins or pathogens in the blood can result in the release of pyrogen, which induces fever by triggering the hypothalamus. You don't need to know this for the exam, but again, having endotoxin inside of your system can result in fever, right? So here's a nice table demonstrating the differences between exotoxins and endotoxins. Again, just to review really briefly, what do I want you to know? Exotoxins are proteins that are coded for by genes. Exotoxins are normally found in gram-negatives, but not always. Exotoxins are heat labile, which means if you heat it up, like boil it, heat it up like really high temperatures, then you can denature those exotoxins. Okay, those are important things to know about exotoxins. And there are different exotoxins, right? Different bacteria produce different exotoxins. Remember botulinum toxin versus anthrax toxin versus Shiga toxin. These are all different toxins produced by different bacteria. Whereas endotoxin, how many different types of endotoxin were there? One, right? Endotoxin means a component of the outer membrane of gram-negative cells. So all gram-negative cells should have endotoxin, right? All gram-negatives. And no gram-positives should have endotoxin, because endotoxin is a major component of the LPS, the lipopolysaccharide. It's the lipid A portion. It's very toxic. It's very it's in nanogram amounts. It's very, very toxic to your system. It's also, here's another thing. Exotoxins are highly immunogenic. That means they spur the immune system to react, and they make antibodies, right? They trigger the creation of antibodies. Endotoxins are weakly immunogenic, meaning they it's a difficult task for your immune system to recognize them. Okay, these are the important things to understand. All right, so that should be the end of the chapter. Just want to thank you for joining me for chapter 14, and hopefully you learned something. Let me know if you have any questions, and as always, I'll see you guys next time.